So, ladies and gentlemen, my name is Sharon Whiteman, and this is another in the series that we're doing for Hope for Lyme patients in May 2022. It's my great pleasure and privilege to host Dr. Gilbert today. Dr. Gilbert is adjunct professor of cell and molecular biology. She's the CEO of Texted Oil that has a mission to get the patients tested so that they can recover quicker. Wouldn't we all want that? With the vast students and researchers mentored by her, she has led large research initiatives focused on directing research for the benefit of patients with better diagnostic platforms for borreliosis and tick-borne diseases. This research led to the development of TickPlex, a completed diagnostic platform that identifies over 15 different tick-borne related diseases, all within a single test. Prior to this, only one disease pathogen was tested out of several tick-borne disease infections. It is costly for the patient to be tested for many individual tests, and if not tested, the tick-borne diseases infections can and will remain undetected. Due to TickPlex, diagnostics of tick-borne disease infections is improved, and patients receive faster access to healthcare and can get personalized treatment protocols so they can recover quicker. Dr. Leona Gilbert continues with Texted Oil only to bring patient-driven research into reality so that their quality of life will improve. And wow, what a great mission that is, Lee. Thank you so much for that.
My pleasure. I'm glad to be here, and thank you for this. Yes.
So, I mean, this is a very complex science topic. It's not actually my forte. And I find the whole testing and diagnosis quite complex for me. But let's start with the gold standard for Lyme disease testing. Can you explain that for us?
Yeah, and it's a great first question, because what is the standard, the gold standard? Well, I can tell you for most of the world, it's in order to get a Lyme disease diagnostic test, as well as to a diagnosis of Lyme disease, usually patients have to go through a two-tier testing phenomenon. And this two-tiered testing phenomenon stems from the CDC, Center of Disease Control testing algorithm that they use. And basically what it is that if you go to your clinician with symptoms, less than 30 days, they will test you either with an enzyme immunoassay test, and I will go over that in a second, or an immunofluorescence assay test. And if you are positive or equivocal, meaning that you could possibly be positive on those tests, you will be tested for IgM and then a IgG Western blot. And again, I will go over those in a second. And so I just mentioned that if your signs and symptoms that you went to present with in front of your clinician was less than 30 days, you get a first test. And if that's positive or equivocal, then you get another test. So it's a two-tier algorithm. But if your signs and symptoms are greater than 30 days, again, the two-tier system is used. Either the first step would be the enzyme immunoassay or immunofluorescence assay. And then if you're positive or equivocal in the results, then they will test you only for IgG Western blot, and I will go over that in a second. But if on the first testing stage with this enzyme immunoassay or an immunofluorescence assay, if you are negative at that first stage, it is recommended by the CDC to consider alternative diagnosis. Or if you have, patients with signs and symptoms for less than 30 days, then consider actually testing them in a few weeks, like 3 or 6 weeks, and attain that convalescent sera, it's called convalescent sera, so that they can, follow if you develop antibodies against the Borrelia. So the standard testing that is used globally is this two-tier system. You have 2 steps and to follow in order to get a diagnosis. And it's interesting because the first step is this enzyme immunoassay, okay, or an immunofluorescence assay, and they are indirectly testing that you have seen Borrelia, which means that you have developed an antibody response. Okay, so you have developed a response, an immune response to the Borrelia. And unfortunately, those tests are only 60% sensitive, which means that there's a hit and miss at that stage. And I say 60% sensitive because that is being reported and published right now. But I do know that there are tests like ours and others that do have a sensitivity of 95%, but that's not widely used. They still use those individual tests that have a poor sensitivity, okay? And then the second step, or the second tier to testing, the Western blots themselves, again, have the same pitfall. They're testing for your response to the Borrelia burgdorferi, okay, and or Borrelia bacteria, your response, so your immune response. And the CDC actually, for this Western blots, or lines as they're called, basically is still an immune system test. It tests your antibodies, but your antibodies are put onto this platform, a blot it's called, and you are to hopefully respond to many bands. If you're testing for IgM antibody, which is supposed to be the first antibody that rises in a response to a pathogen, you have to get 2 of those bands present to be positive. Or if you are being tested for IgG, which is the second antibody that's traditionally raised when you see a pathogen, you have to get 5 of those bands. They have to be present. So it's an indirect response to the body to you actually being infected with Borrelia. And this is the point that what is the standard testing? And I know Nordic countries here don't even deal with, there are Denmark and hospitals in Denmark that don't even deal with IgM testing. They only deal with their IgG testing, and they won't even do an IgG Western blot as a second tier. So they created their own kind of 2-tier system, or their own single tier testing. And then there's some countries that only will deal with IgM testing for children. And we see that in some of our Nordic countries. But then what really is the standard test for Lyme disease? Well, the guidelines are following most guidelines set by the IDSA or from Nice in England, or in Australia, they're following the guidelines. The guidelines 2-tier testing seems to be what they follow with a 60% sensitivity rates. And unfortunately, because they say that it's standardized, but is it really? Some countries, don't go with the 2-tier. Some countries actually, if somebody presents with an EM rash, and again, only about 60% of people present with a bullseye rash once they've been bitten, the site of biting of the tick, some pathology laboratories will test for PCR. So PCR is that direct test. So they will directly test if Borrelia is present. So, and then for spinal fluid, some labs actually test by PCR directly for the bacteria present in those areas, or in synovial fluids or synovial cells, they'll test directly for Borrelia by PCR. Now, so we have direct testing PCR, and then we have indirect testing, which is this ELISA is the basic platform that we test indirectly for Borrelia. But none of those tests are really standardized, because in the United States, they'll use Borrelia burgdorferi sensu stricto as that species of Borrelia that is in the test itself, okay? And they'll use primers for PCR. So these are just little stretches of DNA that actually supposed to specifically bind Borrelia burgdorferi sensu stricto species, okay? And they will use those very specific species-specific testing algorithms to really see if Borrelia burgdorferi sensu stricto is present. And in Finland here, we have 2 different hospitals testing 2 different types of species, and on different sites, if they don't include all the species of Borrelia, how does one feel comfortable that they're testing, for Borrelia per se? So most of the commercial tests that are available, or tests that are available at university hospitals, like here in Finland, they have developed their tests based on 1 species, or maybe 2 species, or a few of the species. But I am seeing right now that in Europe, there are companies that have these ELISAs based tests that are considering the North American strain, if you call it that, Borrelia burgdorferi sensu stricto, and also the other Borrelia strains, European strains, as we call them, Borrelia afzeli and guarini, all three strains in their tests. And that's the point that I want to say, that country-specific species of Borrelia, sure, there's more provenance of Borrelia burgdorferi sensu stricto in the United States, but patients here in Europe have been exposed to that Borrelia because they travel. And that's the point, that afzeli and guarini is finding place in North America. Borrelia burgdorferi sensu stricto is finding place here in Europe because birds are traveling, we are traveling, we're getting exposed even more and more with these species. So the tests need to reflect the nature of the hosts that maintain the Borrelia species, and also us, the people. We travel, we are going to be exposed to ticks, and we're probably going to be bitten by ticks in those areas that have a lot of ticks. So it's important to realize that there is no really gold standard on testing. Many countries prefer to use their own tests. Many university hospitals prefer to use their own test, okay, that is not available to other hospitals outside the country, or to even standardize those tests. So unfortunately, this sounds like a big mess, okay, but there are tests out there that have a better sensitivity than 60% that I just said, and that's the hope, that we actually get into a routine to criticize what is the sensitivity of the test that you're using. Okay, even if it's being used in Australia or United States or Canada or even in Finland, what is the accuracy? What is the sensitivity specificity of that test? And what does the test actually test for? Okay, indirect or directly? And what are the characteristics of the test? Are they using different species of Borrelia? Because if they're only using one species, they're going to miss the other ones. And that's the thing, that's the take-home message of the last 10 minutes of me talking here about testing, is that we need to look and make sure that the best tests are being used, or the tests that have a better sensitivity than 60%, at least 95% should be used to test these individuals.
And how many species of Borrelia is there known?
Yeah, there's many. There's up to about 20 different Borrelia species that are responsible for Lyme disease, and more of those different species are being established more because the hosts are maintaining that Borrelia species more. So, and guaranteed more research in this area will indicate that there's those different the 20 different Borrelia species will be expanded to other different types of Borrelia species. But the prominent ones, though, that we see more frequently are Borrelia burgdorferi sensu stricto and Borrelia guarini and Borrelia afzeli. And these have been seen in Australia, actually, and have been seen in Europe, and has been seen in North America. And it's because people are traveling, and it's also because ticks are traveling on birds as well. So the more time that the Borrelia species can be established locally in the area with the hosts that can maintain that type of Borrelia species, I think we need to catch up, and we need to really accept that locally, this Borrelia species is coming into those areas, is being established, and so forth. Like Borrelia myomoti. I mean, it's now accepted all over the globe, but it started in Japan, and it hitchhiked away over, from a sheep from England to the United States to New Jersey, and then it expanded quite quickly, that tick with that Borrelia species. So it can be really, really fast, and we should just do more research to look at the different types of Borrelia species, that could be in those areas, to really appreciate the frequency, because low frequency will turn into establishment if we just are aware of it, and we protect ourselves, obviously, against tick bites and so forth. So
awesome. And so that must be what brought you to be so committed to this mission, Lee?
I have to say that I have friends, close friends that have been bitten by a tick. Okay, and in Canada and here in Finland, and I started to wonder why certain patients are able to just get over this Borrelia infection or get over this tick bite, and other people can't get over this tick bite because they have all these symptoms. And then I started to really see how these patients are being mishandled. They're not being handled correctly, or they have to be their own advocate to get healthcare. And that frustrates me because yes, there are guidelines that the clinicians follow. Yes, there are these international codes of diseases that a lot of clinicians use to group patients under, so that they can get additional healthcare, or they can be covered under health insurance and stuff like this. But patients need to be handled individually, because the symptoms that one patient may have may not be the similar symptoms the other patients have. And you may say that this is a no-brainer, but in medicine, practicing medicine, you really do not see this a lot. And it's unfortunate because like I mentioned, the first diagnostic criteria for Lyme disease, acute Lyme disease, is an EM rash. Okay, great for you that you present with an EM rash or bullseye rash, but if you don't, you're not going to get treatment. So you have to wait until your clinician believes you or not that you have these objectable symptoms that can put you into the two-tier system. And then by that time, the Borrelia may have actually spread to other parts of the body, and then it becomes a little bit more difficult. We must keep in mind that Borrelia is this low abundance pathogen. A low abundance meaning that it clears the bloodstream quite quickly. So it enters your tissue, your skin tissue, goes into your bloodstream and clears that quite quickly. And that's the difficulty in this pathogen, that it's very difficult to see high amounts of Borrelia in the blood. And if we don't see high amounts of Borrelia in the blood, we can't directly detect it in the blood. We can't culture it, and that makes it a little bit more complicated as to what type of testing would work, because of it's a low abundance pathogen. But Borrelia, as we know, likes to go into these niches, right? Like the knees, right? And that's there. Okay, unfortunately, by it attributing to and infecting the synovial area, we will succumb to arthritis or arthritic symptoms. But it is a site in which we can actually biopsy, and we can actually culture, we can do PCR directly on it, and so forth. So waiting for symptoms like palsy, facial palsy, to occur in an individual and then treat them, I mean, we're past the point that it's going to cause more harm than just believing. And that's why I say, that's why hearing these stories over and over again is why I'm doing this, and why I'm trying to, well, we're just trying to make sure that the research leads us so that we can have some kind of outcome that helps the patients. And I'm not saying that our test kit is the only wonderful test kit. There are great test kits that have a 98% sensitivity specificity, and I advocate for those. Like, we need better tests, better than 60%. We need better ELISAs, better two-tier approaches than using the same 60% sensitivity and specificity. Because I think that even just including the 3 prominent species of Borrelia into the diagnostics brings it from 60 to 98% sensitivity. And that right there is a simple step to do. So let's do it. And let's use those 3 species in the test, like here in Finland, and Denmark and Sweden, and not just one species of Borrelia that they test for, or one antibody as well. Why are some places only using IgG as an indicator, not IgM? Well, then we go into their argument, well, IgM gives us false positives, because I hear that all the time, even from scientists, that they look at the data, well, IgM is false positive, false positive, false positive. But we have to understand, why is it false positive? Is it because of other microbes that they're suffering from, because there's a cross reactivity? Well, then there's other microbes that tell something, right, in the individual. So they are suffering from other pathogens. And our studies have indicated that 85% of the Lyme patients are suffering from other microbes. other microbes that participate in the outcome, the clinical outcome of that individual. That's information that the clinician can take and see, okay, maybe that IgM may be false positive, but there's a reason. There's other microbes there that they have, been infected with, are infected with, and therefore that gives a better picture on how they can handle the patients. Yeah, so it's not this one microbe, one disease paradigm that the clinicians, have been taught to practice medicine with. It is a multiple microbe with multiple disease courses, and it's synergies between microorganisms that cause the disease outcome in the clinical picture of the patient. So we have really we need to change this kind of paradigm, the thinking it's just one microbe consequence. There's multiple microbes that can have a synergetic effect on the patient and how the patient is handling, this Borrelia infection, because it could be many different reasons why. And it's not just because of false positives of IgM and that's causing this kind of confusion. Well, I think the clinicians need to think why is there IgM false positive in their mind, but they don't. They would just want the clear guidelines of how to treat. The two-tier system, IgM, IgG, boom, here, presence, blands or not, and that's it. And unfortunately, people are more complex than that.
Patients are suffering because of it.
Yes, they are. And it's infuriating that they are. And we need to do better. Let's do better.
So do you mind if we clarify? So you mentioned culturing and PCR and the difference between that. And so you mentioned the IgG and IgM is measuring an immune response. Can you explain the PCR and the other types of testing?
Sorry, Sharon, you were sticking.
Regional. Yeah, can you hear me
I'm
now?
sorry. Yeah, you were just paused there for a second. Sorry, to explain the PCR and species specific?
Well, and understanding the difference between looking for an immune response with the IgG and IgM, as opposed to what culturing is. What are these different types of testing?
Yeah, so indirect and direct tests are basically the characteristics of tests being performed on individuals that have a suspicion of Borrelia infection. Directly to test if the Borrelia is present. And under that direct to test, you can culture. So you can take a biopsy, so from your skin or synovial area, and you can put it into culture media that Borrelia likes to grow, and you can culture and grow the Borrelia. PCR also is performed. So PCR, what you do is you like COVID, okay, you can take little strands of DNA that actually bind specifically to the Borrelia species, okay, and it will amplify the area of DNA, okay, on the Borrelia. So it detects directly, and then in the end, you will see a band that PCR did amplify enough. So that's direct. But those primers, those little stretches of DNA that is used for PCR, are usually species specific. Okay, so Borrelia burgdorferi stricto, you have a set. Borrelia guarini, you have a set. Borrelia afzeli, you have a set. Okay, and then you can do, of course, a little bit more sophisticated PCRs that allow very sensitive detection. Okay, but still, you still need to have at least one Borrelia present in your sample. Okay, and that's why in EM rash, I mean, you can really see the spread of Borrelia because in the bullseye rings representing the spread. So good chances that you will be able to see one kind of Borrelia species there for PCR to work. Culture takes a little bit more time, of course, because bacteria like friends. If they don't have friends around, they usually don't replicate so well. But anyways, you can over time culture from EM. And then of course, the indirect test, as I mentioned, is the ELISA, the immunosorbent assay, which basically indirectly tests your antibodies that have been produced in the presence, have been produced when your body has seen Borrelia. And that is the humoral response. So we have that response of the immune system producing antibodies, and we indirectly test for those antibodies as a sign that you have been infected by Borrelia. There are other tests, immological tests that indirectly test that you have seen. I said the antibodies are part of the humoral response, but there is also a cellular response. So immune cell response. So we can test some immune cells like T cells, okay, and test to see if they have seen Borrelia. So basically we will isolate your T cells, your immune cells from the patient and put them onto Borrelia proteins or full lysate, like just break them up, put them on Borrelia. And if they respond, they will secrete cytokines, which is another protein, but that we test for. So that response we test for, and that's an indirect test that you have seen Borrelia. And that cellular test, some labs are using it. It is not FDA approved. It is not widely accepted, but in Europe, more and more, there are CE-IVD marked T cell tests that are being used, and some clinicians are attributing this T cell response as an active infection. And there are more and more reports coming out that these T cells actually are correlating to an active infection. And we need to have more studies on that so that it becomes more and more routine in routine labs. But definitely, I'm sure we'll see more research being published in regards to that cellular response. Yeah. So the Western blots is the last class of kind of the diagnostic testing. Western blots basically are running a Borrelia species down, separating the bacteria based on its proteins that it has. Okay, so you can have a ladder of bands there. Okay, and that's why we call them Western blots, because we're just really separating them based on all the protein sizes that they have in that sample. You can have, it can be very species specific, because you can run many different lines. You can also run just purified protein from the Borrelia, because you can make those artificially, and you can put them at different lines as well. Lines meaning different sizes of protein. And then you put your patient's antibody on top of those Western blots or those Western lines, okay, or immunoblot lines. And you can then read if there is a positive response to those different lines. And that's indicative of these bands. So how many bands do you see if you probe for IgM antibodies? And then how many of these bands do you see if you're probing for the patient's response of IgG against these proteins of Borrelia? And that's why for IgM, you need 2 of the positive bands based on CDC criteria, and IgG, you need 5, because there could be unspecific binding, as people say, unspecific binding of a patient's sera onto these bands. And that's why CDC wants it to be more clear and crisp, 2 for IgM, 5 for IgG, and so forth. And I know that a company in the United States, IGeneX, they have a different criteria of how many bands, and that's good that they're testing this and publishing this, that they could say, okay, well, maybe not 5 is needed, 1 and let's say 6 or 1 or 3, but there may more research on correlating the bands to the different stages of infection is also really good. That should be done, and they're reporting and publishing more and more on those Western blots, and to improve them, because again, it's not standardized, right? Between the Western blot that could be done here in Finland to the Western blot that's done in the United States, they're not standardized, and that's the whole problem of these tests, that they are very, very locally specific, and that's something that we need to do better with, in my opinion.
It sounds like even randomly determined in a way.
Yeah.
You remember we met about, what was it, 4 years ago? We've got to talk globally about this, because it doesn't make any sense for it to be so different when you cross a border.
Yeah, it doesn't make sense. Because the criteria tend to be, and definitions tend to be accepted globally, and the guidelines, but yet the testings aren't globally standardized, which makes no sense. If they're going to use CDC criteria or IDSA guidelines globally, then we need to have a global testing. And that includes, even at a minimum, the 3 prominent species of Borrelia for Lyme disease, in my opinion, of course. And that's something that we're trying to push, this this more awareness of getting standardized testing in to include, the 3 different species. And if locally, if they have their own specific Borrelia, then added to the ELISAs or the PCR protocols that they're using, because right now there are 3 prominent ones, and there are a local acquired Borrelia species, and it's very easy to add those into these test kits. It's not very difficult. Yes, we have to show this safety and efficacy that hasn't changed that diagnostic if you change something or add a new antigen or new pathogen in there. But that's something that we're, commercial test kit manufacturers are used to, and it can be done, and it should be done, of course.
So to clarify, there's
Yep.
Western blot, there's ELISA, and there's PCR, and the higher specificity tests that you're talking about, like TickPlex and others, what are they labeled as?
Labeled, well, they're, oops, sorry, I didn't catch the last one.
Do they fall under those categories, or are they a new category?
No, I think they fall on the category of actually improvements. So they are like TickPlex is CE and IVD marked. It is being sold globally. Okay. And it has the 3 different species of Borrelia. And it has also other things like the persistent form of Borrelia, which is unique. And it also has co-infection, and our TickPlex basic only focuses on Lyme disease, but for tick-borne diseases, our TickPlex Plus has the other co-infections, like the prominent ones, Babesia, Bartonella, Ehrlichia, and so forth. And Euroimmune is another company here that's quite used widely in Europe. Even their basic ELISA has the 3 different species of Borrelia. So I'm not saying we're the only ones. I'm saying that they need to do better. Like, companies or university hospitals or private hospitals that are testing for Lyme disease, just by improving their diagnostic, by going to those test kits and utilizing those test kits that have even the three strains of Borrelia, will definitely increase the detection of patients with Borrelia. And it's not giving those tests are manufactured in ways so that we really see a very specific response, okay, to these different species of Borrelia, and not just an unspecific response, a very specific one. And we are showing right now currently, the utility of this in North America. So again, having the 3 different species, tested for in North America is worthwhile. And we're currently showing that in in Australia, that these patients have traveled, and some patients haven't traveled, and they are testing positive for some species of Borrelia that are present in Europe as well. So it makes while at least the 3 dominant ones test for them, and you'll see that it is a specific response, and it needs to keep or it needs to be put into the practice of medicine, that we're not just testing locally acquired Borrelia, but also, European acquired or global species as well.
Awesome. That's awesome.
Yeah.
And so, obviously the real most important thing about improving testing is that then will lessen patient suffering and improve recovery rates, because early diagnosis is key to recovering from Lyme disease. And so what do we wanted to touch on the emerging diagnostics that are happening right now. Is there anything that you want to cover on that?
So no, I have to say that there are more of these sophisticated PCRs that are being developed, like this digital droplet PCR, that are multiplexing, okay, their PCR algorithms, so that do include also other microbes that are easily detected by PCR. That's something that has been published, ddPCR it's called in short, and we will see that this multiplexing concept, testing for other microbes in the individual, is worthwhile. We see it there. This study came from the United States. And also the other one that's getting attention is an indirect test for a virus that actually infects Borrelia. So that bacteriophage, it's called. Okay, so they're detecting the phage, okay, the bacteriophage, indirectly as a sign that there is an infection, there is a Borrelia infection. That's interesting. Yes, it is an emerging one. It just came out. I think there's one published paper on it, and I'm sure we're going to see more and more of that. For antibodies, there's these sophisticated diagnostic kits that are utilizing PCR and antibodies together to be more specific, because remember, this is a low abundance pathogen. Borrelia is a low abundance pathogen. So there has to be some way that we can see it directly in the blood, because at some stage we should, and acutely we should be able to see it. And so Monica Ambers is working on a project with her collaborators that are using antibodies that are actually attached also to DNA strands that can act like primers, so that you can have a very selected response by binding to certain antigens. And those group up together, and when the little strands of DNA come together, you can amplify that through PCR. So it's kind of like an enrichment step that they're using via these antibodies, so that PCR can be conducted nicely. So that's interesting. NanoTrap is another emerging diagnostic, because it's not FDA cleared yet, but NanoTrap is looking at antigens or proteins coming from the Borrelia in your urine, because some way we need to shed everything as well, right? So that's gaining attention, but of course from the step from scientific published protocol or diagnostic to actually acceptance, FDA clearance or CE IVD mark clearance, or even TGA acceptance. It has to be registered in any diagnostics in Australia has to be registered, obviously, by, what was it again? The ARTG, so the Australian Register of Therapeutic Goods. Yeah, in order for them to be accepted there, of course, more and more published results need to be there. And that's the thing too. I just want to quickly just add that there are these emerging test kits, and one way of getting credibility is to publish in peer-reviewed journals. Okay, but of course, a lot of companies don't do that, and that's frustrating for me, because Eurimmune's test kits, when they came out long time ago, where is the published research on that, so that we could share that knowledge and learn from that knowledge and expand. And I'm not just saying that it's Eurimmune, because they are now coming more and more. Scientists are actually comparing Eurimmune's blots and also ELISAs with their own, like Zeiss and Eurimmune. There's lots of papers comparing those. But I think that what we need on our side too, as a medical device manufacturer of these diagnostic kits, is keep publishing in peer-reviewed journals, so that we can get the information out there, so we can start improving on top of these technologies. And that's, well, I'm getting a sense that that's kind of like what smaller diagnostic companies are doing. They're publishing to get more credibility on their works that they're doing, and so that once they publish, they can use the same results to fulfill the requirements for ARTG or FDA or Health Canada or the European Commission's demands on diagnostics. So
awesome. So
yeah,
what do you think, if you're a sick Australian listening today, what approach, like some won't have doctors and some even with doctors can't get tested, what do you have any advice, or is it just progressing this research?
Well, no, I think hang in there.
Is there any hope? Is there any hope for us?
Yes, there's hope. There are research studies right now being conducted in Australia. Okay, I know that TicPlex is being used in some research that are spearheaded by some clinicians that are really trying to help understand what's going on with the patients in Australia. There are publications coming more and more out of Australia as well, okay, in regards to tick species and pathogen species within these ticks, and also the different types of tick-borne pathogens in patients. And we will see more and more of this. There, of course, there's LDAA that is rooting for these patients, trying to improve these patients. But I know that there are medical device manufacturers trying to get their test kits into Australia, so that they are approved by ARTG, and they can be accessible to, patients. But I understand that. I know it's difficult in Australia right now to get, testing and to be believed by their clinicians, but there are some clinicians there, and hopefully we'll be able to work together to change the situation in Australia, and we will do that with patients and with all these kind of actions that LDA takes, as well as scientists that are taking, publishing about Australia and so forth. And I think through those actions, this will become more and more, while the awareness will be built around this, as well as hopefully better for the patients. And also this whole kind of shift in paradigm where one microbe equals one disease, that shift of paradigm is being seen in some clinicians in Australia that I have noticed. So I'm hoping that they themselves build up that awareness as well, that better testing is needed, and it's not just these guidelines that are not really credible to follow. So
and do you expect, I won't hold you to this, but do you expect anything to be published in 2022, or is it longer away?
I think the early of 2023
Outcomes.
months,
Early.
Some things will be published, especially in regards to some of our initiatives that we are doing, and with some collaborators in Australia. I know that our schedule is the early of 2023, and getting TICPLEX in Australia, of course, that's a goal of mine, and of course we're working on it currently, and let's see how that plans out, because actually, as we know, we're still in COVID time. We are, and I know that these regulators are constantly busy trying to do their job in regards to COVID tests, getting them there. And I know that even in FDA, they are only focused on rapid testing and so forth right now, and other testing are taking the sidelines at the moment, unfortunately. So if we can get over this hump of COVID and move past that with the need and the need of better testing, for Lyme disease and tick-borne diseases in Australia, then I think we will start seeing TICPLEX there. So, but of course, we'll let you know the progress of how that's happening. And I'm just hoping that COVID doesn't prolong it, because it has in other countries.
An alien invasion or something.
Yeah. Well, I know. Sorry, I just wanted to say, I know that there is a lot of this out-of-pocket expenses to get tested, okay, from patients. They can order tests through different, clinical labs like Armin's labs, like IGeneX and so forth. And I understand that it's out of pocket, and I understand that those test results aren't being considered in most cases in Australia, because they're out of country. And that's the thing that also I need to change with, we've been trying to change too, this whole concept that, you weren't tested here, but those are CE and IVD marked products. They are FDA approved products, so this whole concept of global testing, and acceptance of those tests that actually are adhering to regulatory demand by locally, like in Europe, and that they are also, the labs themselves are accredited through this ISO standards, okay, ISO standards that actually are recognized by TGA. Yeah. So I don't that's what frustrates me. I don't.
Yeah.
It is, but it's not tested here. But it is an ISO standard laboratory that you accept, TGA. And those tests are accredited and accepted. Yeah. They fulfill the legal requirements. So why aren't they accepted there? That's the thing that we also need to change, because it's true that there are tests not available in Australia that can be easily accessible through labs like Armin's lab or IGeneX and so forth. And we need to start changing that. And it's not just in Australia, they even say that here in Finland. Well, you weren't tested here, you were tested in Germany. Well, it doesn't matter, because they're CE and IVD marked products. That's the regulatory demand here in Europe. Why aren't you accepting this? And that's the thing that we need to change as well. And we're working on it. We're working on
Yes,
it.
they seem to cherry pick. They cherry pick the standards, don't they, when it comes to Lyme disease?
Yeah, which is crazy because yeah, they say one thing and do another, and I'm like, okay, I don't get this. Yeah.
So I
Yeah.
can see my rural reigning internet is causing problems today. So apologies to everyone. But one of the two other things I noticed today, that there's been some initiatives, and that we're both Canadian originally, and some
Oh yeah,
initiatives announced connecting long COVID and Lyme patients in Quebec. Did you see that, Leona?
I did 15 clinical labs
Yeah, I
that are
know.
going to be established. I'm
Just
like
about fell off
excited
my
about that.
chair.
I know, I know.
I know.
This
I
is
thought of you
great.
right away. Hmm.
Yeah, and
Yeah.
I was thinking that this is such a great initiative to really, and it's not like COVID and lupus or COVID, it's like COVID and Lyme. This is so great because they're recognizing that these long haulers of COVID is pretty much long haulers of Lyme disease, isn't it? And
Hmm.
and this is great. I'm really excited to see what comes out of there, like through publishing as well as their initiatives, because they tell that they're doing this, then good, I'm going to follow it closely and hold them accountable, because I think no, because this is great. I think we're going to learn a lot from long haulers of COVID, because I guarantee the immune system is being, is similar to these long haulers of Lyme disease or chronic Lyme or post-treatment Lyme disease syndrome individuals. Guarantee their immune system is behaving similarly.
Yeah, and definitely Lyme docs in Australia, anecdotally or clinically, are noticing similarities in their, immune cascade issues and
Yeah.
inflammation. So,
Yeah.
the people we're representing today are all victims of an abject failure in health policy in Australia. So, Leona, you've been so heartfelt and so passionate about this mission. Thank you. Is there anything you'd like to leave sick Australians with today?
In that we're trying, we're trying. And I think that we just have to keep going and supporting, those people that are trying and trying to make things better for the patients, and also to reach out if to LDAA or even to me or us, reach out to us if there's anything that we can help with, or I'm good to explaining test results, of course, and maybe what's going on with the symptoms and correlating that to certain conditions and so forth, and just reach out and know that they are not alone. It's a global thing. It's happening here in Finland, it's happening Canada, in the United States, it's happening in Europe, in some countries that the patients are being mishandled, and we just have to know that you're not alone, and that reach out to us, because we're trying, and we're trying to change things. So
Thank
that's
you,
how I would leave it. Yeah.
Dr. Gilbert. Thank you so very much.
My pleasure.